Hello, today we are going to discuss translation and the role that tRNA and several other molecules play in this process. First, translation is the process of converting genetic information in the form of nucleotides to amino acid sequences that produce proteins and other products necessary for the cell. mRNA, or messenger RNA, carries the genetic code for proteins and is made by converting the DNA sequence into RNA. Thus, it is a strand of nucleotides with the bases A, G, C, and U that create codons, which are three non-overlapping nucleotides. The reading frame is made up of a start and stop codon and is the area in the mRNA sequence that is translated. This nucleotide sequence found in mRNA has to be converted into an amino acid sequence. There are 20 different amino acids that serve as building blocks for proteins and can combine in a variety of ways to form different proteins. Amino acids are joined together with peptide bonds, forming a chain-like primary structure. There are 64 different codons, with 61 of them coding for amino acids and 3 serving as stop codons that end translation, seen here. tRNA, or transfer RNA, helps incorporate amino acids into polypeptide chains. The tRNA is made up of several different arms with an anticodon arm that is complementary to a specific mRNA codon and an acceptor arm with an amino acid attachment site. tRNA has to be charged, which happens by the addition of an amino acid to the 3' end of the tRNA at the attachment site. Once charged, each tRNA is capable of transporting only a single amino acid to a ribosome. Translation takes place in the ribosome, which is made up of 2 subunits of rRNAs and proteins. The 2 subunits do not come together until translation takes place. The ribosome has 3 different sites where tRNA can be found: the A site, the P site, and the E site. The A site is where the tRNA enters and recognizes the next codon sequence on the mRNA. The P site is where the peptide bond is formed between the building amino acid chain and the current amino acid. The E site is where the tRNA exits the ribosome. I traveled to Ireland and had to use this adapter to plug into the wall outlets there in order to charge my phone. The plug for my phone did not match the wall outlets in Ireland, but the adapter helped pass along the electrical signal or energy needed to charge my phone. Similar to my wall adapter, tRNA serves as a bridge between molecules and helps to convert messages between 2 unlike systems. tRNA reads genetic codes in mRNA, the A, G, C, and U bases, and converts them into amino acid sequences necessary for cellular function. Lastly, if there is a mutation in the enzyme that helps to charge the tRNA, amino acyl synthetase, or in the arm of the tRNA where amino acid attachment occurs, the tRNA molecule would not be able to act as an adapter molecule. This would cause a disruption in translation and would prevent the translation of a specific codon into an amino acid. Each amino acid has their own specific amino acyl synthetase and specific tRNA or tRNAs. Thus, a mutation in one of these would impact the production of that single amino acid, not every amino acid. Amino acid chains may become shorter as the code cannot be read correctly, and it could lead to disorders due to these mutations. Thanks for watching, and here's a list of my references.